Hi everybody, welcome to the notes. Our notes this time are on basically genetic variation and the things that lead to genetic variation. So as you can see, it's meiosis, sexual reproduction, and mutation. And those are the things that we're going to go over and see how they cause variation. Kind of a guiding question is why don't all children of a family look identical? It's the same parents, the same genes that the parents have, so why don't siblings look the same? Now, early in the development, all the siblings, all people look the same, but at some point, there's differentiation there. There's certain things happen, and so that's what all these processes are going to go through, is how that genetic variation is created. First of all, what is genetic variation? That's literally any change in the DNA. So any change in the sequence. It could be as simple as one base pair. Here we've got cytosine and guanine that are together in that spot, but over here it's adenine and thymine. That's genetic variation. It's one thing that's different. It could also mean whole chromosomes or little parts of chromosomes or chromosomes that are flipped around a little bit. So any change in the gene, the sequence of base pairs, change in the chromosomes, that's considered genetic variation. The process that we'll talk about first is called meiosis. You might have remembered mitosis from earlier in trimester 1. This is something slightly different, and meiosis is the process that bodies go through to produce eggs and sperm. So to compare the two, mitosis is where we took, like if this is a human cell, we have 46 chromosomes. After the process of mitosis occurs, we have two identical daughter cells with 46 chromosomes in them. Meiosis is a little bit different. It starts out with the same amount. We've got 46 chromosomes in that cell, but it goes through 2 rounds of cell division, and then the end result is 4 cells with half the number of chromosomes. In this case, it's 23 chromosomes. Now, the reason that we have to have cells that have half the number of chromosomes is so that we can reproduce. And so sperm have 23 chromosomes in them, and an egg has 23 chromosomes in them, so that when those two things combine, we get our full allotment of 46 chromosomes. And then we start as a zygote, as a single cell. So we all started life as one cell with 46 chromosomes in it. All right, so again, we've got 2 rounds of cell division that create cells with half the number of chromosomes. So if we would have started with 100 here, we'd end up with 4 cells with 50. So it's always, it creates cells that have half the number of chromosomes. So this is a animation that we use the same program with mitosis and protein synthesis and such, but this is on meiosis. So we start in this example with 1 cell with 2 chromosomes in it. And I'll play it through real fast first and see what we end up with. So we started with 1 cell with 2 chromosomes, and we end up with 4 cells with 1 chromosome. Okay, so let's see how that happened. So here's our 1 cell with 2 chromosomes. Through the G1 phase, through the synthesis phase where now it's duplicated, and then through G2, prophase 1. Now this is a cool feature of prophase 1. The chromosomes come together and they get tangled up and switch parts. That's called crossing over, and we'll get to that a little bit later. Spindle fibers attached, they line up in metaphase, and they get pulled apart. Full chromosomes get pulled apart in anaphase, and then telophase and cytokinesis. Now, it goes through another division, so prophase, metaphase, anaphase, telophase, and cytokinesis. So at the end, we've got 4 genetically unique cells with half the number of chromosomes. So these aren't identical. You can see just by looking at the colors that all of them are different. So 4 genetically unique cells with half the number of chromosomes, and those are gametes, or eggs and sperm. All right, so here we are back at prophase 1, okay, of meiosis 1. This is the first prophase, and homologous chromosomes, that is pairs of chromosomes, come together, and they just, their arms get tangled up, parts of chromosomes get tangled up, and you can see it here, they're switching. And so it creates genetic variation because literally one part of one chromosome gets transferred to another, and that one to the other one. So now we have totally unique combinations of genetic information that we didn't have before. Then in metaphase, when they all line up in the middle, there's this thing, a phenomenon called independent assortment. That means they line up randomly. And so you never know which way they're going to line up. So you never know which side which chromosome is going to go to. And that's another way to create variation. And when you're talking 46 chromosomes, that is a ton of variation just by randomly having them line up in the middle. Anaphase 1 and 2. Difference here is in anaphase 1, full chromosomes get pulled to each side. And in anaphase 2, just the chromatids get pulled. They separate at the centromere there, and half of them get pulled here and half of it gets pulled over here. So that's a little difference between anaphase 1 and anaphase 2. So yeah, I know, it's confusing. All right, another source of genetic variation is mutation. And simply put, this is just the order of the base pairs has changed. And we all have like 400 defects the average person has, 400 mutations in the genes. So obviously we are all in this together. We are all completely mutants. So what it looks like, usually like it can be totally fine. Like you can get one change or two changes and there's no disease, there's no effect. Sometimes there is an effect. Ugh. Anyway, here's a scenario, and we're going down the left side here. This is a DNA molecule, and we're just highlighting these three bases. And here's the messenger RNA that's created from that, and glutamic acid is the normal amino acid right there. If you go to a mutation where now this A is different, that means the messenger RNA, the U, that's different. And also it gives you a different amino acid and therefore a different protein, which causes sickle cell anemia. And that's just from one little mutation, one little base pair. All right, the last source of genetic variation is sexual reproduction. And if you think about it, literally, like if you have a kid, it's only half of your genetic information. The other half comes from a different person. And just the fact that that occurs is a ton of variation. So you put some genes, like half of your genes in the sperm cell and half of the female's gene in an egg cell, and those two things combine, and that creates a ton of genetic variation. Okay, so I forgot that you couldn't hear his audio, so I'll go through it. So as the human life cycle goes, the process of meiosis creates gametes. Those gametes combine together again in a process called fertilization, which then makes a zygote. And then that zygote will divide through mitosis to 1 cell to 2 cell to 4 cells to 8 cells, and that'll make a new organism, which will go through meiosis to produce more gametes, and the whole cycle just continues and continues. Anyway, thanks. Okay, I didn't think I ended it really well right there, so thanks for listening. I hope you have a great day, and we'll talk to you soon.